Okay, good afternoon, everybody. I think it is now 15:05. We can begin with our webinar. So welcome to the June webinar of the African Society of Dental and Craniofacial Genetics. Today, I'm sure many of you probably are aware of Inqaba Biotec, but we have a speaker from there. And Dr. Hamilton Ganesan is going to be our speaker today. Previously, we had planned on Mr. Clay Moyler actually giving us the talk, but due to unforeseen circumstances, he's unable to give us the talk. So his colleague, Dr. Hamilton Ganesan, he's the bioinformatics manager at Inqaba Biotec, which is a leading biotechnology company in Africa. He holds a PhD in bioinformatics from the University of Pretoria, along with advanced degrees in biomedical sciences, medical microbiology, and bioinformatics from the universities of Natal and Rhodes. Since joining Inqaba Biotec in 2005, Dr. Ganesan has led numerous projects in next-generation sequencing and played a key role in the conception, design, and data analysis of bioinformatics workflows. He is also actively engaged in the promotion and sales of advanced bioinformatics software, tools such as DNA Star and Advata. Now, Dr. Ganesan, you're going to have to tell me if I said that correctly or not. So welcome. And you are, if you could now perhaps unmute and let us meet you via your video, and you are welcome to take it forward.
So yeah, I was saying to Selma that my video is not working. Can you hear me? Really?
Yes, we can hear you clearly, and we
Okay.
can see your screen very clearly as well. So your
So
video isn't working?
yeah, I dropped my laptop the wrong way, and the lens of the camera got covered, I think. So
All right. No, that's
yeah,
fine, as long as we can see your screen and we can hear you. So you're welcome to take it
okay.
forward now.
Cool. So yeah, thanks, Nobi, and welcome to all the attendees. And I am of the understanding that there's a lot of well, amongst other types of people, there's dental practitioners here. Is that the case?
Well, yes, predominantly it's dental practitioners and mainly individuals that actually work in the sphere of genetics, genomics. So yeah, in the whole omics field,
Okay,
much of the
okay,
research is there.
okay. Well, this is the first time we're speaking to a group from the dentistry fraternity. So we obviously we deal a lot with um, molecular biologists, and now we're delving into the uh, clinical side of things as well. We deal with a lot of doctors, physicians, gastroenterologists, um, and specialists of all flavors. But this is the first time uh, actually uh, doing a training to the dentistry field. So this is, uh, yeah, it's going to be interesting. Look forward to questions. So as you mentioned, there are people that are in the molecular field. So I guess most of this will be intelligible. So for today, I just want to talk about PacBio, and if anyone doesn't know about the company, you're welcome to um, uh, stop and ask questions. But my goal for today is to introduce you to the technology, um, and then explain why the technology is different to others and what's the advantages, and then show you briefly the instruments that run the technologies. Okay, so let's first go into the technologies. So the PacBio's main instruments these days used to be the Sequel 2E system, and uh, and on that system we have a sequencing chip. It's called a smart cell, SMRT cell, and the same with the latest system, the Revio. But we'll get into more details in a bit. But on these instruments, the sequencing happens on that chip. Uh, in terms of what's on that chip, um, it's a it's called a zero mode waveguide. So you can just picture a wafer of silicon, and in that wafer of silicon, there's these very very tiny nanotainers or zero mode waveguides, and um, in these waveguides is where the DNA actually immobilizes for the sequencing reaction. Right? And now there's millions of this indented into the this what we call the smart cell, and in each of these wells there'll be a single reaction. This is another graphic to represent this ZMW. So since light cannot fully pass through um, the ZMW, um, it's a sort of a safe space for your DNA DNA polymerase to advance. So light only penetrates a bottom bit of the ZMW, and that's enough to excite the polymerase into action and then start the sequencing reaction. Um, and each of these wells will have a single sequencing reaction taking place. But of course, there's millions of these across the the smart cell, and so you're getting highly parallel sequencing taking place on that chip. This is another graphic about how that looks. So a single DNA molecule gets. Oh, I don't have a pointer. Uh, oh, can you see my mouse if I do that, Manogli?
Yes, we can actually. So
You
that's
can see.
great.
Okay, so that's fine. Okay, cool. Thank you. So as you can see, these are the nucleotides. They obviously um, labeled with different colors, fluorophores, and these are incorporated into the growing um, DNA strand. And so, like I said, single molecule in a ZMW. Right. Okay. These again are your fluorescently labeled nucleotides. And there's a more chemistry picture of this. So that's your regular DNA and your fluorophore on the end, and that means when this is cleaved, the fluorophore is cleaved, you're left with a native DNA molecule. So what you have at the end is a native or naturally looking DNA molecule, which has some advantages, as we'll talk in a bit. Okay, and then this is just a picture showing you, okay, well, this is how the fluorophores will be, or the nucleotides will be incorporated, and the fluorophores will be cleaved as the polymerase extends that chain, that template DNA. Okay, um, all this happens on this instrument called the Revio. This instrument has um, uh, can actually sequence genome scale genomes. I mean, sorry, yeah, eukaryote scale genomes, like so human genomes, even small genomes, microbial genomes, viral genomes, anything. Um, and we'll see what actually happens on the instrument in a bit. But here's another graphical representation to show you uh, how the polymerase functions to sequence the DNA strand. You get your binding, uh, you get your um, all the fluorophores being flowed into the well at the same time, and only the complementary ones are incorporated. incorporated, it's cleaved, and so you get your emission of your light, and so the process goes. And all happens very fast, though. This is happening in if I understand correctly, each base gets incorporated under 0.5 of 0.5 seconds. So the polymerase works very quickly, but it's actually a biological molecule which was enriched and then bioengineered and slowed down actually, so that when the polymerase action, when that enzymatic action is happening, it's slowed down to a point where we actually can capture incorporation to incorporation. Because in nature, it happens so fast, you wouldn't be able to capture each base being incorporated. So this is a regular molecule that's just been bioengineered to function a little more slower, but it functions in a natural way. Okay, that's that. Um, so what's special about this single molecule real-time sequencing, which is smart sequencing? Well, one thing that really sets PacBio apart is that it actually sequences very, very long reads. And I'm sure most of you know that there's many short read platforms out there, but what's special with PacBio is that it can not just sequence um, um, long reads, but it actually does them with high accuracy, as you can see here. When we retrieve data of the system at the end of the day, the sequences are near perfect. So that means there's no data cleanup that's actually required, and that means any downstream applications would be rendered more accurate because, well, your data is free of systematic errors and all sorts of other errors that is inherent in some other sequencing technologies. Okay, the other thing is that the polymerase functions in a way where it's not biased toward any specific types of regions. So it actually sequences across any genomic region with equal ease, can say, and without bias. So at the end of the day, when you start to do your mathematics or start to do your genome assemblies, you get uniform coverage across the entire um, genomic region that you're studying. And the nice thing, a huge bonus, is that, and we'll see that also in a moment, is that as we incorporate nucleotides, we are also detecting for methylation events on the DNA. And so we'll see why that's important in a moment as well. So here's another explanation of what's happening in that ZMW. What happens when we sequence with single molecule real-time sequencing? So we start off with a native DNA strand, right? Or a template molecule. And onto that template molecule, we add adapters, right? And these are called smart bell adapters, and they form this loop structure on either end. And in that loop structure, we then can anneal our polymerase. And then it starts to unravel this entire molecule. And that's what happens here. We go from this portion being single-stranded to then being, from being double-stranded. And as it sequences, it becomes single-stranded. And each strand is being, or each time the molecule passes over the molecule, we produce what you call a subread, and then another subread. So there's a circular action here, and it keeps sequencing the same molecule. Every time it sequences one portion, that's called a subread, and there's it here. And then it goes and sequences the other side, and that's another subread. So that's the second subread. And then around again, and that's the third subread. So on and on it goes. And not sure if I'm blocking the screen here, but okay. So as we sequence across that same molecule multiple times, the consensus accuracy actually improves dramatically. We do not deliver subreads. We actually deliver what's called a high fidelity read, which is a consensus read of all the subreads. So this is what's called a high fidelity read, and this is what's actually delivered to customers. Um, and this makes for very, very accurate analysis and very sensitive bioinformatics at the end of the day. And I think it's safe to say that this is the data coming off this platform is the best in class data in terms of quality. So as you can see here, with each time that we pass over the molecule, the more we sequence it, the greater the consensus accuracy grows. And so beauty of PacBio technology is that you don't just sequence fragment once or twice, you actually sequence each fragment multiple times, sometimes over 15 times. And then there's that concomitant rise in the accuracy. Um, so even with one, even if you sequence a single molecule 3 times, you already have a 99% accuracy, but we go way beyond 3 passes. And um, we are very confident with the data that's being produced from this platform. Okay, and like we said previously, as we incorporate nucleotides, we also detect um, methylation events. So you see, from incorporation to incorporation to incorporation to incorporation, there's a time difference, and this time difference is um, actually correlates with a methylation event. So um, we can, based on how long the incorporation takes, we can tell uh, what type of modification event is occurring. So the time between that incorporation and that incorporation is called an interpulse duration, and based on that interpulse duration kinetics, we can tell what methylation event was seen on that base. And so this is called a base modification, and this is reported in addition to just your genomic information or your sequence information. And that's a huge bonus. Yeah. So here's that review system again. Here's that smart cell again. And that smart cell um, uh, is loaded onto a single tray here, you can say, or a single stage. But this review instrument is a high throughput, so you can actually have 4 of these uh, smart cells being sequenced simultaneously and independently. So you can see you can have one cell with one project, another cell doing something completely different, and a completely different assay on the others. And so that's amazing flexibility to have on the same platform on the same run. So it's 4 independent sequencing run basically that happens on this. So what type of applications do people um, um, venture into when when requesting PacBio data? Well, obviously we look at assembling large genomes. Obviously, as you all know, personalized genomics is um, always a real boost in interest in having individual genomes sequenced because everyone is unique, uh, and sometimes this changes um, in a specific individual that cannot be uncovered by just comparing to a reference. You actually need to see at the individual level. what their genome profile is. And so yes, people are doing whole genome sequencing for humans on a large scale. We do quite a bit of that. Um, uh, and also because the platform's flexible, we also do viral sequencing, bacterial sequencing, sequence amplicons as well. We also do variant detection. Um, uh, the other thing is we do RNA sequencing. So because the reads are so long, we don't just sequence partial RNA transcripts. We actually sequence the full length transcripts, and you'll see that in a moment. When it comes to complex populations, obviously, talking here about metagenomics, um, so mixed populations from various environments. So yeah, we can cover the full spectrum of applications. Just as an example, the, the because the reads are so long, we have the power to uncover compound heterozygote um, um, uh, cases. And you can see here the certain portion of the genome, the single gene, and we have a specific allele here, and then we have the second allele for that compound heterozygous. But as you can see, because the reads are so long, we can span that entire uh, um, region of interest, and we have the power to pick up both alleles from these longer reads, um, which is not uh, actually possible with short read technologies. So that's one huge advantage of long reads is that you can phase variants into the different chromosomes. Um, it's also used for isoseq. So in other words, we do RNA transcript or transcriptome sequencing just to show you, like for instance, a certain cell types can uh, can have expressed different isoforms. So so that's a single transcript. Um, but when you get back your PacBio data, that entire transcript will be found on a single read, or your single read is the full um, transcript. As you can see here, uh, let's say that is a gene, and those are the exons that were that's the isoform with those exons. A short read would just sequence a portion of that exon, but the long read will span the entire transcript. And so at the end of the day, you can see the full nature of that transcript. And so that's the power of having these long reads. Okay, um, that's in a nutshell what why long reads are important for RNA sequencing. Is that you can uncover isoforms, as and because we can also sequence at depth, you can also do the differential expression as well. There's another example of looking at a specific gene, and because, like I mentioned, the polymerase does not have problem sequencing equally well across the genome, irrespective of the genomic content. And so, as you can see, there's a gene here, and you can sequence this with any technology, obviously. As you can see, like for instance, with short reads, you can get coverage across that entire region of that gene. You see if the short reads are mapped on that region, but then you get portions of the genome where sometimes this drop off. Like if I do whole genome sequencing, sometimes with short reads, there's certain sections that are dark regions, you could call it. Uh, and obviously with whole exomes, those dark regions remain dark regions. But if I do a target enrichment and sequence that entire gene, as you can see with long reads, we actually can sequence across those dark regions too. So and pick up haplotypes that couldn't you, um, you couldn't discern from short reads, and so there's a huge advantage in that you're seeing the genome more comprehensively in a nutshell. Okay. Then when it comes to metagenomes and mixed populations, let's say you had 5 different samples um, represented by each of those bars. You can see how we have the ability to pick up the relative abundances or the abundances of the different groups, different genuses. So you can see how Bacillus has that abundance versus that abundance, and then we can compare that to the Enterococci, to Escherichia. So we have resolution at least on the genus level, but not just on the genus level. We can actually sequence down to the strain level, because on PacBio, because we can sequence long reads, we sequence across the entire 16S molecule, and so because we have this entire 16S gene in hand, we can resolve taxa down to the species and even the strain level. Okay, so that's a powerful way to study complex populations. Okay, and then we don't just sequence 16S amplicons. We can actually do a shotgun approach, meaning we take all the DNA from an environment and treat them as a single library. Do not enrich for anything. Just sequence indiscriminately, and from yeah, well, this is just an example that we can actually detect well, millions of genes in a population in a single sample. And each of these genes, because each of these genes can actually correspond in the end of the day to actual genomes, these little circles represents a genome. So what this is trying to illustrate, that because we have such a depth in data, not only do we have power to predict genes in a mixed bag of DNA, but we can actually assemble that DNA into full chromosomes, full bacterial genomes. So that's also another powerful way of uncovering, what's going on in an environment, which are the predominant, not just which are the predominant genes, but which are the predominant species. So that's just another application. Yeah, like I said, we do epigenetics, so we can tell the various base modifications apart. So this 5MC and 6MA uh, and the other one. There's one more base modification that we can detect. And like I said, there's no special treatment that we do to detect the base modifications. We just look at the interpulse durations, and from those kinetics, which is measured in real time, we can detect what modification is occurring on a particular strand of DNA. That's important because in some cases, the genomic DNA may stay the same. So from person 1 to sample 1 or patient 1, patient B, there's a difference. But even in a single individual, there could be differences between the chromosomes. And you can see here, you have an individual with a differential um, um, uh, methylation profile on these 2 chromosomes, the mother maternal and paternal chromosome. There is a pretty obvious difference which you would not see had you not had access to epigenetic information. And that's again, that's a powerful just bonus information that the platform provides. Okay, and I did show you that big instrument. It was called the Revio, and the Revio has the power to produce 4 human genomes in a single day. So remember, we had 4 stages or 4 smart cells that could be run simultaneously. But that's a pretty big instrument and is out of reach for many people. And so last year, toward the end of last year, PacBio released this instrument, the desktop version, which is called the Vega. This instrument actually runs a single smart cell and produces 60 gigs of output. So you can actually, with this output, you can sequence a single human genome. It runs for a day. You can sequence read links up to 20 kb. You can also detect methylation, as with the Revio, and the base quality is superb, Q30 and upward. Okay, but that's a desktop instrument. Again, it covers the entire spectrum of applications that you would employ on the Revio. And there's it's just at a glance look at the Vega capability versus the Revio capability. And if you look on the left, these are the various applications: whole genome, panels, RNA-seq, microbial, and you can do the same. They both have all these applications on offer, but obviously the Revio on a bigger scale. So for instance, with the Vega, you can do 1 human genome at a time, and on the Revio, you can do 2 genomes at a time on a single smart cell. If you were to do example amplicons, they can offer over 1,000 amplicons on a single run of that, on a single cell. Microbial communities, I can do 64 on a Vega in one run, 124 128 on a single cell on that Revio. But I think the main take home here is that whatever you want to do on the Revio, you can do on the Vega, but the Revio offers it at scale. And let's see, and that's, well, I really rushed through this, but yeah, I'm happy to take questions at the moment. I didn't go into too much detail, but I'm happy to take any technical questions.
Thank you, Dr. Ganesan. Thank you. I think this platform that you've actually spoken to us about now is truly, truly impressive. To be honest, I didn't actually know much about it at this point, and I didn't know the competency of it and what we could actually use it for. So I think just a little bit of background with regards to us being in the dental fraternity. So many of us are sort of specialists. I'm an oral pathologist, and I think there's other dental specialists. But much of what we do in terms of our society and our department back at the university is to do with molecular investigations. For example, with regards to oral cancers and rare diseases. So we also, some of us have, well, a PhD in human genetics. So yes, we do understand the concepts, and we actually do much research in that arena. So for us, we're actually very pleased to know and learn about the platforms that you work with. So if there's anybody that would like to ask a question, please, you're welcome to just unmute and ask or put something in the chat. But while we're waiting, I just wanted to ask you, so if we send something into you, for example, we would like to sequence a genome. Do you
Mhm.
have any reference genomes that you have access to, or do we have to provide that as well, or do we have to find access to it?
Uh, no, no. I mean, most of these reference genomes are publicly available. So yes, we have access, but there's such a vast uh, collection of them. Most
Yeah.
people settle for like HD, your HD38 or your T2T or CHM13. But I mean, we can use if we needed to do the analysis for you, we can use whichever reference you uh, choose.
Okay.
Um,
So with regards to African genomes or sub-Saharan African genomes, the availability of that is also pretty easy, would you say?
oh no, not relative to European uh,
Yeah.
uh, cohorts, you can say, because we are definitely underrepresented. And
Yes.
although there are reference genome sets, there's very few fully complete um, uh, African-based genomes.
Yes,
So,
yes. I think that's the challenge that we face as well, because we want to kind of look at rare diseases, oral cancers, for example,
oh
in the
yes, oh yes,
sub-Saharan African communities.
oh yes,
And one of the things we are busy with is looking at West Africa compared to East Africa. So the only way
yes,
that we're going to actually be able to adequately interrogate that kind of data is if we had reference genomes from those areas.
yeah, yeah. That's the
Suppose
dream.
that will remain a
That's
challenge for a while,
yeah,
isn't
yeah. It will be. I mean, obviously, there's huge efforts from East Africa and MRC 100K,
it?
110K um, initiative to sequence as many African genomes as possible. But I
Right.
mean, 110,000 versus hundreds of millions of individuals is a drop in the
Mm.
ocean. But thankfully, with the more complete a genome can be uh, as a single sample, the more likely we are of uncovering rare disease variants, um,
Yes. Yeah.
uh, even without even a completely closed reference genome.
Mm. Mm.
So that's where the power of the long reads come in.
Yes, yes. I just want to ask a question, and this is for my own interest, and probably others know the answer already. But if we have previously looked at, for example, a syndrome, but not using this type of technology with the long reads, and do you think it would be worth us redoing those investigations um, using this technology, or do you presume we would get the similar outcome?
So we uh, earlier this year was it March? It was a
Yeah.
human genetics conference in Durban, and
Mm.
was it last year at Sun City? It was
Yeah.
that other conference. I forget the name of
The
the one.
SASH,
But
yeah. The Southern Africa.
so yes, yes. So um, one of the keynote speakers was from a guy called Alex Huizen. He is from one of these institutes in the Netherlands, and he basically
Mm.
said they they've been through this process before, where they said, you know what, we're going to go the cheapest route possible. We're going to go the whole exome route. Whole exome route came up empty-handed, right? Then they said, okay, we're gonna jump ahead and do whole genome sequencing, but they did short read genome sequencing, and that also wasn't sensitive enough to uncover, the molecular basis for that rare disease. And then eventually they said, you know what, we're gonna have to do long reads. And so they did the whole exome, they did the short reads, came up empty-handed.
Mm.
However, more often than not, it was the long read based genomes that was the most successful. It had the
Right.
highest success rate, and then they eventually came to a decision where they said, you know what, we're not, because the price of these long read genomes are substantially lower these days. They're going to
Mm-hmm.
skip. They're going to skip the exome, skip short reads. They're just going to go straight to the long reads. And so,
Okay.
so to answer your question, they've
Yeah.
actually they've actually resequenced many samples. Uh,
Right. Yeah, because
and
this is certainly something that we were considering for one of our little cohorts of rare diseases, probably know enamel renal syndrome. And so, yes, I thank you very much for that because now I sort of understand it a lot better, and I think that we can now move forward in the direction that you've highlighted. Selma's just
yeah,
got something in the chat. She said, compared to other high throughput platforms, is it more cost effective? What would you say?
so at this point, PacBio is not the cheapest, not by far. However, um, the utility of the long reads uh, uh, is definitely um, justifies the higher cost. And having said that, I think that
Mm.
we have consistently dropped the price of our long read sequencing. It used to be maybe 5 times the price of a short read genome before. But
Right.
now that gap between a short read genome versus the hi-fi PacBio genome is surprisingly less than you would think,
Mm.
or
Mm.
much smaller. There's a much smaller gap, much cheaper. There has been a few chemistry advances in the last 6 months, meaning
Mm.
they have boosted the output of the the current cells, which in effect means you're paying the same for more data. So again, that's another factor that brings down the cost per genome. So
Right.
so I invite you
And
to maybe request a quote, and you can see for
yeah.
yourself that the price is not as high as you think it would be.
Right. And if you say that it yields better results or more productive results, then certainly it would be, worth
Yeah,
us
yeah,
paying that extra.
yeah. But we
That's
obviously
certainly
uh,
something we I think we are engaging and um uh s- and we also do know that Inqaba Biotech does, for example, the bioinformatics analysis as well, or assists
yes,
with it.
yeah, yeah, yeah,
And
yeah,
for
yeah,
us that's sort of just still evolving in those fields, that's a big benefit.
yeah. But we have platforms that's extremely user friendly,
Mm-hmm.
and so we can put bioinformatics in your hands. We have systems where you can upload the raw data and do the filtering and uncovering of the variants on your own. You can put in your HPO term or uh, your own clinical definitions into an analysis, and the analysis would be automated and pull out any variant that's even remotely related to the condition you're searching. And so this is something that's done
Okay, that's
online,
good to know.
and you can look it up. I'll give you the reference is genyx.com, uh,
Mm-hmm.
and so you can
If
see
you could
what
maybe
it's
just put
capable.
it in the chat, if that's okay.
Do I have chat here? Oh yeah, okay. Um, so
Ah, there
and
it
we
is.
yeah
Okay.
yeah so we're dealing with a lot of cases where surprisingly it's resequencing
Mm.
of older samples because
Yeah.
it's an important case,
Mm.
and 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 the several of these researchers have found um, the answers they were looking for with the long reads.
Yes.
And in some cases they didn't even need to do whole genome. They actually were looking to phase variants in a single gene, and they couldn't do that with short reads. And so they just actually did amplicon sequencing, and
Hmm.
that was in many cases the the silver bullet, so to speak.
Yes,
Um,
yes, yes.
but we'll
So
help you. I mean, if you have a case, we'll tell you what's the best approach.
yeah, yeah. Okay, no, that's fantastic because, we are still sort of evolving in the field. So having direction when we engage with the company also helps us. And you did
Yeah,
mention you do single cell sequencing as well.
yeah, we do offer single cell as well. Yes.
And that would be maybe advantageous in terms of, let's say, different cancers.
Yeah, yeah,
Okay.
yeah, for sure. Well,
Oh,
yeah, it depends on your application, but that's also growing in momentum as well.
okay. So I think
Yeah.
just one more question from me before we let you go.
No,
Um,
I'm not in a hurry, but yeah.
I just wanted to know, you said that in terms of epigenetics, you're able
Mhm,
to also identify some abnormalities. So I know, for example, like in FASD and things like that, there's no sort of test out there.
mhm.
But is it worth, could one actually identify a condition like FASD? If we
You
send
mean is
you, oh,
um
sorry, it's fetal alcohol
yeah,
syndrome.
yeah, I know. I was thinking with Cape Town people. uh, um well, um I'm trying to recall if there was a study to examine the clinical utility of uh, of genomic data for FASD. And
Oh, okay.
maybe it's been done. I'm actually not sure. But if you wanting to profile, well, the child versus the mother, uh,
Mm,
you could definitely do that comparison in terms of methylation with PacBio.
okay. No, because we have one of our colleagues, Dr. Iman Romani, is probably on this call at the moment. Um, she's just completing a PhD in FASD.
Hmm.
And so perhaps she sort of know more or have more questions in terms of that. So if there isn't anything else from anybody else, we thank you very much for all the information. And going forward, you certainly will be hearing from us. And thank you for stepping in. I think your colleague was unable to make it today, so thanks for stepping in today. And I certainly learned a lot. I'm sure many people on the call did as well.
Yeah.
Oh,
What was another?
oh, there's one more
Ooh,
question. How easy can the vagal system be? Ah, there we go. What do you think?
well, um, the PacBio systems are RUO instruments, research use only. But I mean, there's always an adjacent clinical, as a clinical lab that's leveraging the PacBio technology for some clinical purpose. But is there like a registered diagnostics sequencer from PacBio? No,
Not at
no,
this point.
no. But I mean, they are using it to process clinical samples and do the customized sequencing work that
Mm,
that's not being done, on standard other standard platforms. So
right. So it's basically research based at this point.
at this stage, yeah,
All
yeah,
right. Okay, so I mean that's kind of understandable because you would think that you have to do the research before you can actually put something out there and offer
yeah,
it.
yeah, yeah,
So I,
yeah,
I'm sort of understanding where it is at this point. All right,
yeah.
so thank you and everybody. Oh,
Well,
there's another
just one more
person
thing.
that says it's
So
very informative. Yes.
So I've been hopping on the long read technology, but there's actually maybe for another time. There's PacBio also has released um, a short read uh, platform, uh,
Mm-hmm.
which is called the Onzo. I'm not sure if this link would work, but I'm not sure if that even is a link. But there's an Onzo system, and that also has well, everyone knows Illumina, right?
Yes.
Because Illumina has been pretty much the only sequencer around for the longest time. But
Yeah.
PacBio has released an instrument called the Onzo, which produces Illumina type data, which is your same short read paired in, but at a higher quality. And so, if you guys are needing more information about that, we can talk about that at a different stage.
Okay, no, that's great. That's good to know, actually. Uh, so yes, we will certainly be in conversation. And I think for me, one of the good things is that we're able to have a conversation with you
Mhm.
and you're able to tell us what might be the best option moving forward, which I think as clinicians, we don't always know that.
Mm.
Um, so we are at 15:56. We're almost at a full hour. So thank you very much. And
Pleasure.
if
Yeah.
there's no more questions, we shall log off and meet again at another time.
Okay. Thank you for the
Thank you, everybody.
privilege. Yeah. Okay. Bye.
Bye-bye.